Good evening, everybody. Very good evening to all of you. Welcome to the Brahmastra Series session, everyone. Good evening, sunshine. Yeah, Sampurna. Good to see you here. Preeti, Ashwarya, Ramdas. Good evening, good evening. Good evening, everyone. Yes, good evening, everybody. Welcome to the session. And it's good to see all the evening batch students here. I'm very sure that you people are already busy and still you took out time to attend the session. Very good. I really hope that after this 1 hour you will find this time well spent and you will find it fruitful, and it will help you in understanding the process of translation even better. So today, as you know, as the topic also is mentioned, we are going to talk about translation and post-translational mechanism. So I have taken out some questions which are commonly asked, which has been asked in previous year CSIR papers. So all the questions that we are going to discuss in next 1 hour are only from this translation process, and also the towards the last part of the translation, right? Because last week also we had one session where we discussed the translation related questions, but they were for the initial stages of translation, like the codons which are required, amino acyl tRNA synthetase, how it charges the different tRNAs, how it recognizes the different tRNAs that needs to be charged. So those are very important. Amino acyl tRNA synthetase, okay, anything related to that is extremely important. At least that has been the trend in last 5 years, I can say. Every session, one question from amino acyl tRNA synthetase alone has been asked. So you can see how important that part is. So anything related to that, you should know. So today we are going to talk about we are going to discuss the questions which will come in the like from the process, okay, initiation, elongation, termination, as well as the post translational modification. So those are the questions that I'm going to discuss now, and these questions are all from previous year CSI papers. Okay, are you all ready? So I hope all of you have gone through the basics as well before coming to the session, so that there is no confusion in the process. And now we can, because once you have the concept, on the basis of that you can question, okay, but just from question don't that will be a wrong way of studying, that from the question if you are try to understand the concept, okay, so that will be wrong, okay. And the question is very precise, so you might understand the base the thing that the question is talking about, but apart from that the entire concept you will not know. So that's why it is important that you learn the concept and then take questions. Okay. All right. So, very first question: which one of the following codon is used to code selenocysteine? And this question was in this June 2023 CSIR exam only. Which codon? Straight away, it is asking which codon. So in yesterday's classroom session, okay, so we were discussing this, right? I gave you a brief idea, so that's why I thought that I should include this here so that we can discuss a little bit more on that as well. Yes, yeah, most of you have given the correct answer. So the answer is UGA. Okay, UGA. No, Anupam. UGA. Okay, so UCC, Anupam, first of all, remember selenocysteine. Selenocysteine is not an usual amino acid. Selenocysteine is added as it's a modified amino acid, right? So this modified amino acid, this selenocysteine is coded from one of the stop codon. So UGA, UAG, and UAA. These are the three stop codons. Okay, UCC is not even a stop codon. So remember that selenocysteine is a modified amino acid which is coded by one of the stop codon. Okay. Now, which stop codon it is? It is UGA. So it is UGA that codes for selenocysteine. Okay. So as we were discussing in last class, and I will tell all the students also who are not joining the classes, that this UGA, how it codes for selenocysteine. So you might think that UGA is a stop codon. Then how does the cell knows that now I have to put a selenocysteine in place of UGA, or I just have to terminate the translation? So that the cell recognizes by some signal I was telling you, right? So that signal will be a stem loop structure which will be formed, that is called as SECIS element. Okay. So that is this SECIS, it stands for selenocysteine insertion sequence element. Okay. It stands for selenocysteine insertion sequence element. All right. So this selenocysteine insertion sequence element, so that basically this stem and loop structure, that actually gives a signal to the cell that for this UGA sequence, it has not, it doesn't have to terminate the translation. Instead, it has to incorporate a selenocysteine at this place. Okay. So the mechanism is little bit similar only in prokaryote and eukaryote. This stem and loop structure use SECIS will be there, but this SECIS elements position in prokaryotes and eukaryotes is different. In case of prokaryotes, this SECIS element will be present in the close proximity of the UGA codon, whereas in case of eukaryote, it will be present at the 3' UTR region. Okay. But in case of eukaryote, what happens that we will do, and you might have studied also that in gene regulation, that in case of eukaryotic DNA, the DNA can bend. Okay. So because the DNA can bend, that's why it can bend like this here, and then this segment can come close to the codon UGA. Okay. So that will that will recruit the other auxiliary proteins which are required to add selenocysteine in response to the codon UGA. Okay. Now, because of the, did you all understand what signal? So, Anupam, in yesterday's class, you were asking about, right? What signal? So, do you understand? This stem and loop structure is the signal that gives this, that will tell the cell that now it has to, in place of UGA, if it is there, followed by this particular stem and loop structure. So, that gives the idea to the cell that now for this UGA, it has to add selenocysteine and not terminate the translation. Okay. Is it understood? Right. Now this is just the signal. Okay, apart from that, there are factors which are required. So in case of eukaryotes, this factor is SBP2, okay, which stands for this SECIS binding protein. Okay, and then it also requires the elongation factor that will support the elongation with the help of selenocysteine. Okay, so that is called as small EF, that is eukaryotic elongation factor, SEC. Okay, selenocysteine, and also it will require the selenocysteine specific tRNA, a tRNA that carries selenocysteine. Okay, so these are the other requirements, and in case of prokaryotes, it will require the homologous proteins corresponding to this. Okay, so when selenocysteine needs to be incorporated in place of UGA, so the cell not only have to have this SECIS element in the DNA, but along with that, it should also have the genes that will code for these elements which are required, these auxiliary proteins which will be required for incorporation of selenocysteine. Okay. Now, moving on to next question. Now, this is also a very interesting question. The amino acid arginine is encoded by 6 codons: CGU, CGC, CGA, CGG, AGA, and AGG. Assuming inosine is not an option in the tRNA anticodon, what is the minimum number of tRNAs that would be sufficient to read these codons? So after the exam, I remember last session, many students came and asked me that what will be the answer. So to answer this, what concept will you have to use? Yes, everyone, can you quickly respond? To answer this question, which concept you have to use? Yeah, Rimi, I'm actually looking for the answer, like not this question's answer. I'm asking like to answer this question. What concept? Like this question is based on which concept? Which concept will you use to answer this question? That how many tRNAs will be required? Okay, or will be sufficient. The minimum number of tRNAs that will be sufficient to recognize these 6 codons. No. Yes, Preeti, absolutely correct. The Wobble hypothesis. Wobble hypothesis, the concept of Wobble hypothesis we have to use, right? Because how many codon a particular tRNA can determine, that is identified with the help of the nucleotide that is occupying the first position of the anticodon. Right, so the nucleotide which is occupying the first position of anticodon will recognize how many codons it can recognize. Right, now for Wobble hypothesis, what we know that at the third position of the codon and the first position of anticodon, the pairing can be little unstable. Right, so if it is X, Y, and X here, so here it will be Y and X, and here this pairing. Okay, so this can be little unstable, provided, what is provided, that this is the mRNA which is 5 prime end here and 3 prime end here, and that's this one is the tRNA whose 3 prime end is here and 5 prime end is here. Okay, so this position is called as the Wobble position because this is the place where the pairing can be unstable. Correct? Okay. Now we know that in tRNA, how many anticodons that particular tRNA will recognize will depend on the nucleotide that is occupying this position in the tRNA. Right? The first nucleotide of the anticodon. Okay. So there we know that there are 3 different types of anticodons. Okay, one set is the one which recognizes only 1 codons each. So what are they? So that will happen when at this position, okay, the first position of codon, if that is occupied by A or C, because A can pair only with G and C can pair only, sorry, I'm so sorry. A can pair only with U and C can pair only with G. Okay. So if this is the nucleotide that is occupying either A or C, then that particular tRNA can recognize only one codon each. Okay. Because anyways, there is no flexibility in the other 2 positions. Flexibility is there only in this position, the Wobble position. Okay. Now there are tRNAs which can recognize 2 codons each. Okay. When the first nucleotide of the anticodon is either U or G. Because if it is U, then it can pair with A also and it can pair with G also. Okay, and if it is G, it can pair with C also, and it can pair with U also. Okay, so basically what does that mean? So if you take this two, okay, if we take these two codons, right? So if this is the codons, means the 5' end is this, 3' end is this, right? So what will be the anticodon? So I will write it down, 3' here and 5' here. Okay, so U pairs with A, G pairs with C, C pairs with G. Okay, so it can be ACG, right? But what we know that if A is coming at this position, then there is no flexibility. It can pair with U only. Okay, but we know that if it is G here, okay, then there comes flexibility. Where if it is G, okay, so if at this position it is G, so G can pair with U also, and G can pair with C also, right? So both of this codon can be recognized by the anticodon having this particular, can be recognized by the tRNA having this particular anticodon. Do you all follow? Everyone, do you follow? So what does that mean? If you have a tRNA with this anticodon 5' and this is 3', it can recognize both the codons CGU and CGC. Okay, right. Now again, this next two, if you check, okay, again they are differing at the third position only. The other two position is same. Okay, but this other two position, if it is so again it will be C and G. Okay, this will be the 3' end. So at the 5' end, what nucleotide will come? U can come, because U can pair with A also, G also, right? So if this is the anticodon, okay, so the anticodon, if it is UCG, then it can recognize both the codons CGA and CGG. Okay, and what about the third set, AGA and AGG, right? So here also it is A and G at this third position in the codon. So A and G both can pair with U. So that means if we have a tRNA with the anticodon UCU, it can recognize both the codons. Clear? So minimum how many tRNAs we need? We need only 3 minimum tRNAs. Do you all understand? Understood. So I will show you this image also. So this is just showing only with one anticodon. Okay. So as you can see, this anticodon UCU. Okay. So it can recognize the codon AGA as well as AGG. Okay. Because U at this particular position, at the Wobble position, can pair with A and can pair with G as well. So same tRNA can recognize two different codons. Okay, now definitely that is only possible when they both are coding for the same amino acid. So two synonymous codons. Okay, so before we go ahead, I am very sure that you already know that the dates, the tentative dates for the CSIR exam has already been announced, that it will be between 10th of December to 16th of December. And that's why Biotechnica is here to launch the Raftaar batch. which is our revision batch, okay, high speed revision batch. That's why the name is Raftaar. And we always every time, like before 3 months, we launch this batch. So now the batch has been launched, and it is starting from 11th of September. So along with that, definitely you're not only getting classes, you will get all these different things that will help you in your preparation, that will make your preparation more fruitful. And this 3 months, okay, that will be the best time of your life, where you will study hard, you will understand concepts, and you will qualify your CSIR exam with your hard work and biotechnical support. So if anybody has any question, any doubt, please call us in this toll-free numbers. Rimi, that's okay if you have not attended the previous class. Anyways, we are doing question discussion only, so that is okay. So if you understand the concept of translation, there is no need of there is no continuity from the class. Okay. So in any case, if you are not able to understand any concept, please ask me. Right. Next question. During each cycle of chain elongation in translation, how many conformational changes does the ribosome undergo that are coupled to GTP hydrolysis? You have to think about what are the steps that are happening in the chain elongation in translation. And then you have to answer how many different changes ribosome undergoes, which is coupled with GTP. Yes, I'm waiting. What do you all think? Okay, yes, I can see that all of you are confused between 2 and 3. Okay, so chain elongation step of translation can be divided into 3 steps, right? So, okay, so in this case, what are the steps involved? Okay, so first step will be what? First step is recruitment of amino acyl tRNA at the A site, right? Next step is attachment of mRNA with the small ribosomal subunit. Oh, no, sorry. I'm talking about elongation. I'm so sorry. Next step number 2 is peptide bond formation. And third step is translocation. Okay. Now, does it help? So, during the recruitment of amino acyl tRNA, so when the amino acyl tRNA is brought to the A site, 1 GTP is hydrolyzed. So in case of prokaryotes, so what happens? It is EFTU, which is bound with GTP, that will escort the amino acyl tRNA to the A site, right? So, and once the amino acyl tRNA, the codon anticodon base pairing happens, this GTP will be hydrolyzed, right? And then EFTU will leave. Okay, so that is one place, one conformation change that happened because of GTP hydrolysis. And next one, Peptide bond formation does not require any GTP. The next GTP will be used for translocation when the ribosome will be, so that will be done by EFG. That will move the ribosome to the next codon on the 3' side, okay, which will utilize GTP, right? So how many conformation change the ribosome is undergoing during elongation? It will be 2. I'm sorry, answer is 3. Option 3, but it is 2. Clear, everybody? Okay, moving on. Right. So this is also one type of question which is very commonly asked in CSR, which is related to this part. So there will be some graph or some experimental, the result of some experimental analysis will be given to you. Okay. So you have to analyze the graph. Okay. And on the basis of that, you have to choose the answer. And also you have to consider the information that is given to you in the in the question. Okay. So for this question, ribosome prepared from a bacterium was fractionated by sucrose density gradient centrifugation, okay, to separate 30S, 50S, and 70S populations. Okay, so that information is given in panel 1. Okay. So when the ribosome preparation was incubated individually with either elongation factor G, that is EFG, or newly identified protein X, or GTP, the profile remained unchanged. Otherwise, likewise, no change were seen in the profile when ribosome preparation was incubated with EFG plus GTP or protein X plus GTP. However, when the ribosomal preparation was incubated with protein X, EFG, and GTP together, it resulted in change of the profile. Okay, which showed a decrease of the 70S peak area and increase in the peak area of 30S and 50S. Okay, so what is this peak area is showing? So basically this peak area is showing the concentration. Okay, so more is the concentration of that particular element, more peak area is coming. So in panel 1, if you see, the peak area for 30S, 50S, and 70S is given. Okay, so when you see that, what do you see? That there are least number of ribosomal units are present in 30S form. Least number of then more than that, it will be the larger subunits present, and maximum it is the whole ribosome that is present. 70S means it's the whole ribosome, right? Now, what is happening that when you are incubating this same preparation, okay, with EFG or X or GTP alone, okay, so I will just write it down. So preparation plus GTP, preparation plus protein X, or preparation plus EFG. Okay, in all these 3 conditions, no change in the graph. Next, what you are doing, now you are incubating it with 2 elements at a time. So preparation plus GTP plus EFG, preparation plus GTP plus protein X, or preparation plus protein X plus EFG. Still no change. Right? And at the end, what it is given? That when preparation is incubated with EFG, GTP, and protein X, then there is a change. Okay. Now, what change is happening? The 70S concentration is going down, and 50S and 30S concentration is increasing. The peak area under 50S and 30S is increasing, but the peak area under 70S is decreasing. Do you all agree? Whatever I have written here, do you agree? Is that the same thing which is given in the question? Right. So this is very important, guys. This is also one step that you should do. So when this analytical kind of questions or some experimental setup is given in the question, now it's very important that you take out the important points and write a note. Okay. That will help you analyze. Otherwise, just words, 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 you will not be able to understand anything. When you take out the important things, write it in your own words like this. Okay. Then it will help you understand what is happening in the experiment. Okay, fine. Now let's go back and see. Okay, so what will be the reason for that? So choose the option that defines a correct conclusion from the observations. So A says that protein X is an anti-association factor which functions in presence of EFG and GTP. B says protein X is a dissociation factor which functions in presence of EFG and GTP. C, protein X binds GTP, and D, EFG is known to bind GTP. Hence, it has to be it can be concluded that the effect of GTP is through EFG and protein X does not bind GTP. Okay, only one is correct, right? So, what will be the most probable one? Okay, so this one or this one, they cannot be true. Why? Because that does not give us any explanation about why 70S concentration is going down and 30S and 50S concentration is increasing. Okay, so straight away eliminate C and D. Now A and B, out of that both can be correct. Okay, one is saying that, so we for sure know that protein X, it can work only when GTP and EFG both are present, right? Because when alone it was present, it was not doing anything. It reduced the concentration of 70S. Basically, that means what? That the hollow ribosome was dissociated in its subunit components, right? So this whole was 70S, so that was dissociated into 50S and 40S, and that is why the peak area under 50 and 30S, so that is why the peak area under 30 and 50 increased, but under 70 decreased. Okay, right? So out of A and B, what can be correct? So it A says that it is anti-association factor. Okay, so anti-association factor can prevent the further association. Okay, but can it break the existing associated form? The answer is no. So it can break the associated form when X is a dissociation factor. Then only it can dissociate the hollow ribosome into its subunit component. Okay, so answer, most probable answer will be option B. Easy, everyone. Yes. So did you see that when this kind of question is there, if you just read, if you keep reading this, you will not understand and you will keep getting confused. So always take out the important stuff, write it, okay, and then analyze. That will be much easier. And when it is easy, you will reach the conclusion also much faster. Okay, next question. During elongation step of protein synthesis, translocation moves the mRNA and peptidyl tRNA to tRNA by one codon through the ribosome. Translocation in E. coli involves GTP and EFG. However, in vitro translocation can take place independent of GTP and EFG. Based on these observations, the following hypothesis can be made. Okay, so what is the situation given? That we know about translocation. It happens as a part of elongation step, and that makes the ribosome move. Okay, to the next codon, right? Now, the translocation in E. coli, it involves GTP and EFG. EFG is the translocase it's the translocase factor. So EFG does the hydrolysis of GTP, and that gives the energy for the ribosome to move. Okay. But what we see that in vitro, when we are conducting translocation, then ribosome can translocate independent of GTP and EFG. Okay. So these are the four hypotheses which are given. So what do you think is the most probable one? Fine. So A says that molecular mechanism of translocation in vitro is completely different from that in vivo. Second says translocation activity is independent of GTP hydrolysis. Next, translocation activity is completely dependent on GTP and EFG. And next, translocation activity is inherent in ribosome. However, the rate of translocation in vivo is enhanced significantly in presence of GTP and EFG. Okay, so let's see all these hypotheses. Yes, all of you have given the correct answer. Okay, so first hypothesis says that molecular mechanism of translocation in vitro is completely different from in vivo. Okay, so this is less likely, okay, because it is the ribosome only we are talking about. So definitely ribosome, the nature of ribosome, whatever is the nature of ribosome, it will sustain. Okay, there might be little difference, okay, but it cannot be so drastically changed. All right, so the in vitro or in vivo, again, when we are talking about protein translation by ribosome, the process will be similar only. Okay, so it completely different, not true. Second, translocation activity is independent of GTP hydrolysis. Okay, so if it is independent of GTP hydrolysis, then why inside the in vivo, why in the in vivo condition it requires GTP? Okay, so that is a question. So this seems doubtful. Next, translocation activity is completely dependent on GTP and EFG. Now, this also is not true. Why? Because if translocation is completely dependent, then how come in vivo condition translocation is happening without GTP? Okay, but when we see D hypothesis, which says that translocation activity is inherent in ribosome, however, the rate of translocation in vivo is enhanced significantly in presence of GTP and EFG. So that seems to be correct. Okay, now do we have any option of B and D? No. Right? So, and D seems the most exact explanation of the process, that ribosome in absence of GTP also can translocate, but the speed of that will be much less. So inside the cell, when we want to increase, when there's a need to increase the speed of translation to make the process efficient, that is when GTP is required and EFG is required, so that the process of translocation can be much faster. Okay, so answer will be option 1. And there's a research paper also. Okay, so there's a research paper also in Google also, you can check where the experiment was done. Okay, the experiment is mentioned where like in vivo condition without GTP, without EFG, it was checked like how if the ribosome is moving from one position to another. Okay, so that research paper is there, and definitely you can read it and it will give you more information. But the thing is, I want to show you that if this kind of question comes, okay, and very sure, like I will say that 90% of the people will not be aware with the experiment, okay, the exact experiment. So then how you can analyze the question itself to get to the correct conclusion. Okay, so yes, true, it can, like you might not be 100% sure, okay, but at least you can reach 90% surety. All right, and now in that case, in 90% surety if you have and you are attempting this question, it should be a good calculated choice. All right, moving on, next question. This is about puromycin, which is one of the antibiotic which kills the cell by terminating the translation process. So puromycin is an antibiotic used to inhibit protein synthesis. Given below are few statements about the antibiotic. Okay, so from this section of translation, this translation inhibitors, okay, puromycin, streptomycin, chloramphenicol, diphtheria toxin. Okay, so these are all translation inhibitors. Okay, questions from this part do come. Okay, so you have to remember the mechanism. All right, so in that case, it will be better if you make a chart with the antibiotics name or the toxins name and what is the action it is doing or how it is stopping the translation, inhibiting the translation. If you do like that and revise it, so all the antibiotics or all the translation inhibitors you will get at one place. It will be great for revision and you will understand their mechanism also faster by doing a comparative study. Okay, so puromycin, what does it do? Okay, so puromycin, it resembles amino acyl tRNA. Okay, and it enters the ribosome through the A site. All right, now not only that, puromycin, what it will do, it gets incorporated in the growing polypeptide chain as well. Okay, and then that chain which has got puromycin incorporated is released, is terminated prematurely. Okay, so puromycin, the answer will be puromycin resembles amino acyl tRNA and binds to the A site of the ribosome. All right, and then it will result into premature termination. Into premature termination. Okay, and then this puromycin, okay, so it is found to inhibit growth in gram positive as well as gram positive cells as well as animal cells and insect cells. Okay, so puromycin inhibits both prokaryotic and eukaryotic protein synthesis. All right, because it resembles the amino acid tRNA which is present in both prokaryote and eukaryote, so thereby it can enter the A site of both prokaryote and eukaryote and it can get incorporated in the chain and it results into premature termination. Right, so this is the way how puromycin is working. Okay, so you see this is the chemical formula of puromycin. Okay, so when puromycin is incorporated, so puromycin will enter the ribosome through the A site. Okay, not only that, it will also form the peptide bond with the amino acid chain. Okay, and that will be a puromethylated peptide and that will be terminated prematurely. Okay, see, it is released. So it is prematurely released. Okay, and this puromycin, it can it can terminate translation in prokaryote as well as eukaryote. Okay, so that's why the answer will be option 3, D and F. Both D and F conclusions are correct. Next question. During translation in prokaryotes, when ribosome reach the termination codon, the termination codon is recognized by class 1 release factor, RF1 and RF2, leading to the release of polypeptide. A second class 2 release factor, RF3, facilitates the termination process. Which of the following statements regarding the mechanism of action of release of factor is incorrect? Okay, now this is also one important thing. So nowadays this type of questions comes very frequently. Okay, that you have to select the correct option, correct hypothesis, correct combination, or even incorrect statement or incorrect combination. Okay, so you have to be very careful to answer these questions, or else what will sometimes happen that when you are in too much hurry or when you are not properly focused, so even though you know the answer, you will take a wrong question. अगर इनकरेक्ट को करेक्ट आपने ये पढ़ लिया तो आंसर गलत कर दोगे, है ना, आंसर आते हुए भी गलती हो जाएगी, right? And that will be very very costly. Okay, and let me tell you that that hurts even more than the questions that you don't know and you did not attempt. जाना हुआ क्वेश्चन जब गलत हो जाता है ना तो बहुत ज्यादा दुख होता है ना, तो इसलिए जब आप ये ऐसे वाले क्वेश्चंस देखो तो मेक श्योर ठीक है, ऐसे तो वो लाइक क्वेश्चन पेपर में आप देखोगे इट विल बी ऑलरेडी हाईलाइटेड इन बोल्ड और इट विल बी इन कैप्स, बट यू हैव टू मेक श्योर दैट यू रजिस्टर दैट इन योर माइंड. Okay, what kind of statement you have to choose, correct statement or incorrect statement. Yeah, Lisa. So, without any stop codon, also puromycin can terminate the translation. Yes, of course, that is how puromycin work. Okay, so that is why, like when you, if you are using puromycin to puromycin as an antibiotic, so it will kill the cell. How it will kill the cell? By terminating all the protein synthesis. So all the trans proteins that will be formed, okay, the primary structure of the protein, they will all be prematurely terminated. They will all be truncated. So the cell cannot survive without the protein, and the cells will die. Okay. Right. Now, coming back to this question on release factors and the termination process of translation in prokaryotes. So, before going ahead, the answer is this. So, let me show you the chart here. Okay, so what happens is that whenever a termination codon is there, okay, then no tRNA in the cell is there which can identify the termination codon. So now the RF class 1 factors will come, RF1 or RF2, depending on what codon is there, what termination codon is there. Okay, now RF class 1 factors bind. Okay, then two things can happen. One thing is it will immediately hydrolyze the polypeptide chain and it will release. Okay, and then RF3 GTP will bind. Okay. The other thing is that before the release of polypeptide only, RF3 GTP binds. Okay. And then the hydrolysis of the polypeptide chain happens. Okay. Now, how does the hydrolysis of the polypeptide chain happens? That the RF1 factor, okay, they will have a GGQ motif. Okay, GGQ, these are the glutamate, and the Q amino acid. Okay, so they are present. The GGQ motif is present, and that will result in the hydrolysis of the polypeptide chain. Okay, and so after that, what will be the situation? That GTP, so once the polypeptide is released, okay, now the GTP, which is bound to RF3, okay, the class 2 release factor, now that will be hydrolyzed. Okay, and as that get hydrolyzed means GTP gets converted into GDP. When that get hydrolyzed, RF3 dissociates. Yes, sorry, I'm so sorry. Yes, the GGQ motif is glycine glutamine. Thank you, Vigneshwari. Yeah, Achal, it's not glutamate, it's glutamine. Rimi, in Biotechnica, we, like in the classes, we solely follow English only. But yes, if the students are having some doubt, any specific doubt, so we can have some small discussions in Hindi as well. Or else we also conduct like special sessions can also be conducted for those students for who needs this like to understand the concept. They need Hindi. Okay, but in Biotechnica, we always encourage students to get in the habit of talking and understanding English, because ultimately when you are going for research, there is no escape from English being the global language. You have to be confident in, and you have to be proficient in English. Okay, so that's why we try to encourage people here to understand the concepts in English only. So the mode of teaching is majorly English. All right. Yeah. So what I was saying, that what do you see? That when GDP gets hydrolyzed, sorry, when GTP gets hydrolyzed to GDP, okay, then and PI, okay, then RF3 is released. Okay. So the question there was about So you see the statements, okay? So option 1 says class 1 release factor decode the stop codon, while RF3 is a GT paste that stimulates recycling of class 1 release factor. Absolutely true. Okay. Now let's leave the B statement and see RF1 and RF2 share a conserved sequence of GGQ, which is essential for the polypeptide release. Absolutely right, because this GGQ sequence only will catalyze, will stimulate the hydrolysis of the polypeptide chain. Then in option D, RF1 and RF2 individually possess another stretch of trinucleotide sequence which are involved in the recognition of termination codon. Yes, that is correct. Okay, that is why when the termination codon is there, this class 1 release factors can come and enter through the A site. Okay, so this statement is correct. Now coming to the B statement, it says that free RF3 has higher affinity for GTP than GDP. Okay, now see what happens when RF3 is bound by GTP. Okay, then it binds with ribosome. Okay, and when RF3 is bound by GDP, then it releases from ribosome. Right? So what does that mean? That RF3 have more affinity for GDP. Free RF3. Free RF3 means RF3 which is not bound to ribosome. So free RF3 has more affinity for GDP. That's why it is released. It moves away from ribosome. But when RF3 is bound with GTP, then it has more affinity for ribosome. That's time it goes and attaches. Okay. So free RF3 has more affinity for GDP. All right. So this statement is incorrect. So we have to choose the incorrect statement. That's why this is the answer. Okay, see Ashwarya, RF1 and RF2, what are they? They are the class 1 release factors, right? So when the termination codon comes, just make clear, yeah. So when the termination codon is here, so that termination codon will be recognized by this class 1 release factors. Okay, so how will the class 1 release factor recognize? So this class 1 release factors, they will have a specific amino acid motif which will recognize the termination codon. Okay, and that is why it can come here at the A site and enter the ribosome. Okay, moving on to next question. This is also about the translation inhibitor, that is chloramphenicol. So chloramphenicol is a broad spectrum antibiotic which inhibits protein synthesis in prokaryotes. Given below are a few statements regarding the mode of action of chloramphenicol. Chloramphenicol inhibits peptidyl transferase activity of ribosome. Chloramphenicol can be used to treat moderate to severe infection because mitochondrial ribosome are not sensitive to chloramphenicol. Chloramphenicol binds to one of the domain of 23S rRNA. Chloramphenicol competes with binding of E site of tRNA. Okay. Now let's see what is the role or function of chloramphenicol. So as you can see in the image, what does chloramphenicol do? Chloramphenicol will come and attach with the larger ribosomal subunit at the peptidyl transferase center. Okay, and then it will stop the peptide bond formation. Stops peptide bond formation. Okay, that is why how chloramphenicol will work. Okay, so it will stop the peptide bond formation. So whatever stage, so at that particular stage, the protein synthesis will stop, will be blocked. Okay, so that is the mode of action of chloramphenicol. Going back, right? So chloramphenicol inhibits the peptidyl transferase activity of ribosome. True. Okay. Now you remember we have talked about this. So who or which is the element in ribosome that catalyzes this peptide bond formation? That is done by the 23S rRNA, right? Which is a part of the larger ribosomal subunit. How chloramphenicol is inhibiting this peptide bond formation? By binding with one of the domain of 23S rRNA. Okay. Now, B is not correct. So chloramphenicol is used to treat moderate to severe infections. Okay, so the thing is that chloramphenicol can be used, it's a broad spectrum antibiotic. It can be used in mild to medium conditions. Okay, why? Because when chloramphenicol is used in higher concentration, then it will affect the mitochondrial ribosomes as well. Why? Because chloramphenicol binds to 50S ribosomal subunit. Our mitochondria, the mammalian mitochondria, also have 50S subunit. So at higher concentration, chloramphenicol can go and bind there as well. And in that case, that it will inhibit the translation process in mitochondrial ribosomes also. Okay. So higher concentration, it is lethal for eukaryotic cells as well. Okay, and this competing with the E site is for sure not correct. So answer will be A and C only. Okay, so I'm just a little hurrying so that we can do one more question for last 4 minutes. Okay, so next one, a nonsense mutation in the protein coding region of an upstream gene of a group of genes in an operon often leads to depletion of the downstream gene products. This is a classic example of the phenomenon of polar effect of the mutation. Following statements are being made about this phenomenon. Okay, so what is this polar effect? The polar effect talks about that when a particular mutation happens. Okay, so suppose this is 5 prime and this is 3 prime. Okay, in the mRNA. So if a particular mutation happens here and that affects the transcription and translation of the genes which are present downstream of that site. Okay, that is called as polar effect of a mutation. Okay, now it is saying that suppose a nonsense mutation happens. So what are all the mutations that can have polar effect? Nonsense mutation means a coding codon gets converted into a stop codon. So if that happens, then premature termination happens. And if that is the case, then whatever proteins, whatever genes were present downstream, that will not be expressed, right? And that happens majorly in case of operons, where multiple genes are placed together under the same control elements, right? So if this is the control element and multiple genes are present, let's say gene A, B, C. Okay, now within the gene A segment, there is a mutation which is happening, a nonsense mutation happening. Okay, then the translation will stop here, right? The transcription will stop here, or the translation, if the nonsense codon has been done, nonsense mutation is there, so the translation process will stop. Okay, so these genes which are present downstream of the mutation site will not be translated at all. Okay, now based on that, read this 5 statements and see which are most probable. Yes. Okay, see, so what happens is that when the termination codon is upstream in the ORF, okay, then it depletes the ribosome that travel down to the downstream ORF, because it will stop the ribosomes there, it will stop the translation process, and ribosomes will not even go to the downstream part. Okay, and if that happens, then it helps in loading the Rho factor. Okay, and that results into premature transcription termination, but it will be the Rho dependent termination. Okay, so it results into Rho dependent transcription termination. All right, and that is also one of the mechanism of how the the concentration is maintained. Okay, concentration of various proteins are maintained in the cell. Okay, so this is like for a nonsense mutation. Okay, so if a nonsense mutation happens, then the ribosome traveling to the downstream part of the mRNA will reduce, and that will allow the row factor to be loaded, and that leads to row dependent transcription termination. Okay, now if there is a nonsense suppressor tRNA. Now what is a suppressor tRNA? Okay, see, suppressor mutation means a mutation, a second mutation that suppresses the effect of the first mutation. Okay, so here the mutation was what? That a nonsense codon was created, and nonsense codon means it does not have any tRNA that recognizes that particular codon. Okay, now a suppressor tRNA will be what? A legit tRNA, but with a mutation in its anticodon segment, so that now it starts recognizing a nonsense codon. Okay, that means in place of that nonsense codon, now termination is not required. In place of that, a legit amino acid can be added. Okay, so that will be a suppressor, nonsense suppressor tRNA. Okay, so presence of the suppressor tRNA reading the nonsense codon generated by the mutation will diminish the consequence of polar effects. Okay, so what was the consequence of polar effect? That translation was stopping. Okay, so now if the nonsense codon can be recognized by the suppressor tRNA, that means one amino acid can be added and translation does not have to stop and it can continue. So answer will be statement C and E. So I just gave you I added an image of a suppressor tRNA also for you to get the idea that this is the suppressor tRNA, right? So this is the 3' end, this is the 5' end. Okay, so when this tRNA, the tyrosine charged tRNA is there, so it has the anticodon GUA. Okay, now this GUA cannot recognize the stop codon UAA. Okay, so therefore translation stops. Okay, now suppose a mutation happened in the tRNA coding for tyrosine, so that the anticodon now the tRNA is having is UUA, not GUA. Okay, now this UUA anticodon in the tyrosine can recognize UAA on the mRNA. So now this stop codon or the nonsense codon is no longer a stop codon, and corresponding to it a tyrosine can be added and the protein translation can continue. Okay, the more on this the suppressor mutations and all we will also talk about in unit 8 as well. Okay, in unit 8 as a part of mutation, this suppressor mutation is there as also. Okay, but this is how a suppressor mutation, which is also called as this nonsense suppressor mutation that suppresses the effect of a nonsense mutation, okay, can be used to diminish the effect of the polar effect of a nonsense mutation. All right, so we are already past 6 o'clock, and tell you the truth, I did not even realize 1 hour is over, and I still had so many questions to discuss. But yes, we can see it again in the next session or in any other coming session. Okay, so before ending this, again, I would like to show you that our Raftaar batch is going to start very soon from 11th of September. If you know somebody who wants to enroll, or if it is you who wants to enroll, please do not hesitate and contact us in the toll-free number. I hope you had a good session, and you will keep continuing watching all the Brahmastra series at 5 o'clock. See you next time. Bye. Keep practicing, keep studying, okay? And clear all your concepts. Thank you everyone for joining. Bye-bye. Have a great evening.